I'd like to thank organizers for giving me this opportunity to introduce our study of comparison of de-duplication methods used by WHO GLASS and Japan nosocomial infection surveillance in the surveillance of antimicrobial resistance, which was recently published in PLOS One. WHO GLASS aims to enable international comparison, analysis, and sharing of AMR data by encouraging countries to submit their national AMR surveillance data of the target pathogens in a unified manner. The target pathogens and their specimen types are shown here. The submitted data are used for creating reports that tabulate and visualize, for example, proportion of non-susceptible isolates and frequency of antibiotic-resistant infection per 100,000 tested patients for each antibiotic and each target pathogen. According to the guide to preparing aggregated data file for GLASS, it recommends first isolate of a given species or one isolate per patient de-duplication stratified by specimen type. It requires selection of only the first isolate of a given bacterial species per patient per surveillance period per specimen type, and furthermore, per gender, age group, and infection origin stratification. De-duplication is a major issue in the surveillance of AMR in order to make epidemiological sense of aggregated data after removing repeated isolates. The de-duplication method of GLASS is simple, feasible, and consistent with recommendations of CLSI. However, no study has reported a comparison between the de-duplication method from the GLASS system and any other method. According to a previous study, difference in de-duplication methods can cause up to 10% difference in resistance rate. So the question is, how about the de-duplication method of GLASS compared to another more complicated method? Here, we conducted comparison using the national AMR data from the Japanese National Database of Japan nosocomial infection surveillance, so-called JANIS. More than 1,000 hospitals are already participating in this surveillance, which collects all routine bacteriological test results, including both culture positive and culture negative samples of all sample types. The data are automatically converted in each hospital laboratory and uploaded to a cloud system every month. In total, data of millions of samples are uploaded per year. After the data upload, the cloud system conducts various data processing and creates both national report and feedback report to each hospital as benchmarking to evaluate and motivate infection control in that hospital. We recently started to analyze the comprehensive national AMR surveillance data for various purposes, as demonstrated in the recent publication in Journal of Hospital Infection. In the data tabulation, JANIS uses a method that removes repeated isolates of the same species isolated from a patient within 30 days, regardless of specimen type, in order to count patients from which the species was isolated, as colored in red. This rule is applied because a subsequent bacterial strain culturing after 30 days usually corresponds to a different episode given the average length of a hospital stay less than 30 days in Japan. But in order to count isolates, it still distinguishes isolates that showed change of antimicrobial resistance phenotype, for example, from susceptible to resistant for any antibiotic, as different isolates that are counted up. Therefore, the de-duplication method from JANIS is conceptually different from that of GLASS. This slide shows a summary of the differences in the de-duplication methods from GLASS and JANIS. Time period in which the de-duplication is conducted is surveillance period, for example, 12 months in GLASS, whereas it is 30 days in JANIS. GLASS accounts for specimen types, but JANIS does not. On the other hand, GLASS does not account for AST results, but JANIS does to count isolates. Based on the understanding, in this study, we exported all bacterial data consisting approximately 8 million samples from approximately 1,800 Japanese hospitals in 2017 from the JANIS database and tabulated using either the de-duplication method of GLASS or JANIS. Here, you see comparison of the percentages of resistance for each antibiotic stratified by outpatient, inpatient, and specimen type. In E. coli as a representative species tabulated using the JANIS or GLASS methods. The dark blue was calculated using the JANIS method that does not account for the AST results, but just uses the 30 days rule to count patients. The light blue was calculated using the JANIS method that can account for the AST results and count isolates. The red was calculated using the GLASS method. Overall, there was little difference in percentage of resistance to any of the surveyed antibiotic. The difference among the three groups was always less than 4%. Comparison between the dark blue and light blue, namely the two JANIS methods, showed that the dark blue kept the resistance values more similar to the GLASS outcomes colored in red than the light blue. This is because the light blue conducts an additional selection and counting step to distinguish isolates with change of antimicrobial resistance phenotype obtained by AST. But both dark blue and GLASS method do not. The difference is, however, small and at most 3%. Next, the number of tabulated patients whose blood and urine cultures were taken is shown here, stratified by outpatient, inpatient, and age group. In the former, both the total number of outpatients and inpatients de-duplicated by the GLASS method, colored in red, were approximately 140,000 and 200,000 patients more than those included using the JANIS method, colored in blue. The result was similar in the latter. The discrepancy between the JANIS and GLASS probably comes from the fact that GLASS counts the number of patients infected per anatomical site, whereas JANIS only considers the site of the first occurrence of the pathogen, but ignores the others. When the difference was examined between the three age groups, it was largest in the higher than 64 years old group, irrespective of outpatient, inpatient, and specimen type. This may be because older patients tend to have multiple specimen types, such as blood, urine, and sputum. As a publicly available product of the present study, we have created an Excel tool that stores and can analyze the national data in Japan, tabulated using the GLASS de-duplication method. It can be used by simple mouse clicking to generate stratified antibiograms according to GLASS. Furthermore, we have tabulated the data from 2011 to 2017 for each prefecture using the de-duplication method from GLASS. The data are publicly available and were incorporated into the One Health platform website, in which each antibiotic can be individually evaluated for its geographical distribution regarding percentage of resistance among the prefectures and its annual trend. In conclusion, the two tabulation methods did not show considerable differences in terms of the tabulated percentage of resistance, and this study significantly deepened our understanding of data tabulation in GLASS, which helped us to develop this publicly available national data tool and website. Thank you very much for listening.